Only once I ask you to be on time, Professor Rubin. You want me to go back to the slides when I congregate you? Later on at home, okay. So, origin is from the United States, according to the USDA. And later on you see the platinum, the road distribution, every yellow. Even in Hawaii and some other places you see Central and South America, Africa, East, India, Vietnam, Australia, and should have one yellow dot here, Israel. The generated GPIF niche model shows the ability of distribution of platinum to other parts of the world. Very frightening. And you see all the reds are probably the best condition for platinum to grow. And this is according to Climax, while we made our PRA for EPO. That means that almost everywhere, if you take out the very northern countries, are suitable for platinum distribution. So Israel is very nearby, and we have. Our neighbor should be in alert, Turkey, Greece, and then all Mediterranean, of course. And they can go up to, you see, Italy, Romania, Croatia, Spain, France, and some other countries. The distribution in Israel is not a very big country, as you know, but we have it a lot. You can see that it's in the Boom area. It's the Bet Shean Valley. This is the Lake of Galilee. This is the Jezreel Valley and few places along the seashore. So it's Mediterranean climate, not too heavy rains. Let's say temperature can be from the Bet Shean Valley for about 40 on summertime to 6 or 7 on winter, and it's grown very, very nicely. Now, a few slides to convince you that we are talking about one of a hell of a weed. This is platinum in date tree. You see young date tree and a platinum very nearby. We irrigate on summertime a lot of watering to the date, so it's excellent environment. Then you can see alfalfa, and it's spread along the cultivation and mowing and get it. So if you have 1, 2 in the first year, and this is a 2, 3 years growing season, so it continues spread all along the field. This is in cotton, in the very orange Jezreel Valley. You can see how many platinum. This is a cotton for, not for growing for crop, but for genetical modification or genetical improvement or what, like this. In sunflower, of course, sunflower, it's a 180, more than 1.5 meter to smile. Okay, so platinum in sunflower goes up all the way for the photosynthesis, of course, and flower and produce seeds and remain very excellent situation. This is in field tomato. You know that we are growing field tomato for industry, ketchup and all kinds of. So you see platinum in field tomatoes. That's why we decided to make a competition experiment, and we'll talk about it later. We decided, and we counted according to the growing, that we have about 16 plants of tomato in square meter, and about 36 plants of platinum in square meter. So we develop a competition as should be in different proportions. Of course, 100% platinum, 75 platinum, 25 tomato, 50-50, 25 Protenium, 75 tomato, and 100% tomato. And it looks like this in boxes. Each one of them, it's 1/4 of a square meter. It means 4 like this are like 1 square meter in the field. This is the beginning after planting. This is sometime later on. And this is almost in the end. The Protenium goes up all the screenhouse right in the Faculty of Agriculture. And you can see the picture has been taken in August, in the summer. The result. The Protenium and tomato growth, and you can see the blue is the mean fresh weight. The red is the mean dry weight, and you see all the planting ratio, tomato 25%, and tomato 50, and 75, 100, and you see different according to the amount of Protenium in the experiment. You see the optimal competition X should be in the 50-50 if you share a plant and a weed. So this is not really similar to the maximum ability, but you can see that Protenium is a bit different. But you see 100 and you see that the fresh weight decreased along the competition. The dry weight is acting very similar to the fresh weight as should be, and we see a decrease. This is the influence of the Protenium on tomato fresh weight in several planting, and you see what the difference, that every percentage show or decrease the fresh white or dry weight of the tomato plants because of the Protenium in the competition. I decided, after cutting the Protenium on the soil level, to see, even if it's an annual, to see the ability to regrowth. And believe me, it was a miracle. I mean, it has regrowth after 45 days after cutting the plant in 0, and you see the shoot dry weight. You see statistically, when you have less Protenium in the beginning, or even 75%, you still have a biomass is a second growing from the same mother plant. This is the number of the new shoots from the average from every Protenium in the experiment. Sorry. The one,
we need to put it
no, this is
full, yes,
yeah,
we need to put it on
full
full
screen.
screen.
Okay, put it in full screen, please.
Yes, and then you have to go to your, okay, which was, tell me where to stop.
One after the last. Okay, another one.
Yes?
Yes. Continue,
Yeah,
continue,
yeah,
continue,
yeah,
continue,
yeah, yeah.
continue, continue.
Yes?
Another one? This one. No, this one. Okay. Sorry, I push the back instead of the head. So the number of the new shoots in the experiment, in the 35%, we have about 5 new regrowth shoots, and in the 50, almost the same, and a decrease to the full Protenium in the competition, but we have between 4 and 5 new regrowth shoots after cutting into 0 level. It means that even in annual weed, if you had at least one eye of viable ability to regrowth, can grow almost to be as a second year of a plant. Okay, in conclusion, I hope that I am on time. The presence of Protenium styroforos at all planting ratio caused a significant reduction, about 30% of a tomato shoot biomass per square meter. The Protenium plant gained about 25% increase in shoot biomass at all combination with the tomato plants. Intraspecific competition of Protenium Sterophorus was stronger than its interspecies competition. Patellum Sterophorus has rigorous ability after shoot cutting of the soil level, and we decided and we concluded that Patellum Sterophorus is a real threat as a competitor to field tomato. I hope that this is written okay now.
It's right.
It's right, Talia?
Yes, it's right.
Barakalo.
So thanks to you for the presentation. We still have some time for one question. Yeah.
תשאל בבית. No, sorry. Sorry.
Why did you choose biomass instead of yield, fruit yield, as your metric for competition?
It's a hard question. It's not that I decided biomass because first of all, Patellum, you mean yield of the tomato.
Yeah.
But the conditions, this is not real on the field. So even if I tell you that it will be like 16 tons per hectare or something like, it's not real because it was on a container, on a medium that it was the best to achieve the most biomass. In the field, it's a bit different. And of course, we took all the weeds in between and left only tomato or Patellum. We knew that it would be something like a question, but we should continue for a competition in a field situation. This is a big problem.
Is biomass and yield well correlated in tomato?
The biomass and yield, if you don't have any pest, disease or something like, or parasitic, it is.
Is there another question for Tuvia?
No problem. We don't have time.
Yeah. Go ahead.
Well, in the distribution map of the Hysterophorus, I saw it was all across the continents in many different points with different climate suitability map, but in reality, that species wasn't in Spain, but it was in Sweden. Is there an explanation or?
The ability of the Patellum, the flexibility, it's huge. Of course, it could be an optimum. The fair, humidity, high temperature, different climate between the summer and the winter. But we have in Israel, we've been in Ethiopia, Africa, a huge inoculation of Patellum. We've been in India, it's the most infested weed in India. And they didn't say anything about allergenicity and something that this is European weed said is not a working group. It's highly allergic plant like ambrosia. Some of the people, for example, he touched Patellum and he became red and ran away with 2 legs, not like now. This is highly allergenic weed. So when we made the climate, the PRA on Patellum, we figured out that almost everywhere, unless I don't know, part of Siberia, Russia, would be inoculated by the Patellum. The only need was the introduction, and come from after the first or second year, and you have got the population.
So thank you, Tuvia.
Thanks a lot.
So just before introducing the next speaker, as you saw, Tuvia was so excited to present his presentation that he didn't let us the time to present ourselves and the session. So as you can see, we are in the session of wheat crop interaction, and I'm Stéphane Cordeau. I'm from France, from INRAE, and I'm an agronomist. And I will let Barbara to present herself and introduce the next speaker.
Yeah, so I'm Baraibar from the University of Lleida in Spain. And so next speaker is Andrea Cavalieri. And I want to say for the people online, there's no, the other room doesn't have the facilities to have to see the online presentation. So this is the presentation in this room is the only one that you can see online. So I'm sorry about that.
Good afternoon, welcome to everybody. My name is Andrea Cavalieri and I'm a postdoc in the Weed Ecology and Evolution group of PONIV at the University of Copenhagen. And plant-soil interaction, new perspective in crop weed competition is the title of my talk of this afternoon. I would like to start with a general overview. During the recent year, there has been a growing interest both from private industry and academia in screening and selecting microbial community, beneficial microbial community, to be used to increase plant growth, plant health, and plant fitness. These products are commercialized as biological and biostimulants. On the other hand, there is a lack of research on the possible effect that these biologicals can have altering the rhizosphere microbial community of the plant, and so on plant-plant interaction, and so with a question about what could happen for crop weed competition. These represent both a challenge and an opportunity for the weed research community to start investigating and developing research on crop weed competition and a new perspective for crop weed interaction. We note that weeds interact and compete with crop for resources available in limited supply, water, light, and nutrient. And so in this framework, it's really important to understand and describe crop weed competition to be able to develop research on weed biology and weed ecology. So far, much of the studies on plant crop competition has been addressed measuring the relative reduction of above and below ground biomass, developing competitive ability and intensity indexes. So we could argue that plant biomass is still the main currency in plant ecology. But we also know that plants, when they're competing with neighbor individual, they compete also biologically and physiologically, and that is having an effect on the rhizosphere microbiome. With rhizosphere microbiome, we want to describe the thin layer of soil attached to the roots. And this layer of soil is rich in secondary metabolite, in compounds, in microorganisms. And during the recent year, they had lots of attention, mainly for increasing tolerance to abiotic stress, improving growth and development of plants, solubilizing nutrients, so make the nutrient more available for the plants, and protecting against pathogen. Still, little is known about the effect and the fate of the rhizosphere microbiome when plants are competing. So far, at least on my knowledge, the first paper addressing this topic has been a paper from 2017, so quite recently as a research area, from Hortal et al. In their work, they describe the competition between two species. These are two perennial species. They're not really agricultural species, but still. And they check, they test how the microbial community is changing when plants are growing in monoculture, so two plant species per pot of the same species, or in mixture, two plants per pot, each one belonging to the different species. And the results are that when growing in monoculture, each species is able to develop its own species-specific rhizosphere microbial community. When they're growing in competition, the weaker species, the microbiome of the weaker species resemble that of the most competitive. But we also know that competition is density dependent by definition. And we know some of the outcomes that could happen when plants are competing at increasing density. If they're seeds, they remain dormant, reduce growth and so plasticity, or increasing self-thinning and mortality. Still, we don't know what's happened to the rhizosphere microbiome when the competition increase and density increase. So we develop a more comprehensive studies using 5 different plant species growing in monoculture and in a 5 species mixture. And we check the effect of density on the rhizosphere microbial community in the monoculture and in the 5 species mixture. The experiment was developed in a greenhouse using mesocosm, big pot, 48 liter, filled with soil from the organic research farm of the University of Copenhagen in Tostrup. The 5 species are co-occurring weeds in natural environment or agricultural environment. Cornflower, dragonhead, Italian ryegrass, Plantagopsilum, and white clover. And we check, as I mentioned before, when the rhizosphere microbiome and the biomass, when the plants are growing in monoculture or in a 5-species mixture. These are the these are not. These are the density, high enough to induce mortality in the plants. And we collect the above ground biomass, total, and sorted by species, and the rhizosphere microbiome. The above ground biomass were used to calculate the relative competition intensity. So on the y-axis, we have the index. It means that for a positive value, it means the interspecific competition is stronger than intraspecific. So the plants produce more biomass when they're growing in monoculture than when they're growing in competition with the other species. At the opposite, negative value, it means that the plants produce more biomass when they're growing in competition with other species than when they're growing in monoculture. So this is the trade-off between inter and intraspecific competition. And these are the 5 species. Different colors represent different density. And we focus, although we analyze all the data, and all the microbiome, we focus anyway our analysis on these 2 species. Cornflower, because its competitive ability, or competitive performance, let's call it, increase with increasing density. And white clover, because it changed from being a discrete competitor at a low density and a weaker competitor at a higher density. We collect the rhizosphere microbiome. Just briefly, with a small shovel, we lose the soil, we pull out the plant, we shake gently just to have only the rhizosphere still attached to the plants. We cut the roots and the rhizosphere, we put in an Eppendorf, we snap in the liquid nitrogen, and we did the DNA analysis, quantification, and then we send all the sample for NGS analysis, next generation sequencing. And we calculate the different indexes. The first things that we wanted to address is the impact of plant species and density on the community composition. So for doing that, we use the beta diversity, Bray-Curtis dissimilarity. And just to explain you, the value is ranging from 0, so the sample are not dissimilar, to 1, the group of sample are dissimilar. Different colors represent different plant species, different shape represent different density. I hope it's possible to appreciate on the screen. It's not really easy. But anyway, we had the monoculture and the mixture. The permanova of the monoculture shows a significant effect for plant species, not for density and not interaction. And only 2 species cluster separately from the other. And that's our white clover and Lolium Italian ryegrass. The other species that cluster together, they are all dicot. So this is not really results, but it could be interesting to keep it into mind for a future perspective for crop weed competition, how anyway the species cluster by functional group. Much more interesting are the results from the mixture, in which we had plant species and plant density effect, both of them significant. Made exception of Plantagopsilum, the other species cluster together. So it's exactly the opposite. When plants are competing, they develop their own species-specific micro community, so they hold their own. As I told you before, although we have the analysis of all the species, I'm going to just focus on the most competitive and the less competitive species. And main results is that for the dominant species, cornflower, both in monoculture and in mixture, there were no effect of density and of growing and of treatment, growing monoculture or in mixture. while for the white clover, there were a density effect I forgot about it. This is the richness, so the number of species, and this is the Shannon index, so the diversity. And, but there were no effect on the mixture for both of them. But having a closer insight of how it's changing the relative abundance for T. repens, we saw that there is a shift from being reliant on rhizobium in monoculture, start instead recruiting other microorganisms from the rhizosphere microbial community, still able to fix its nitrogen, but it's not rhizobium. So for its own competitive advantages. This is just the main results for summing up. So we had an effect of intraspecific competition were different and larger than those of intraspecific competition. For the dominant species, there were no effect for intra or intraspecific competition and density effect, while for the weakest competitor in the monoculture, it was mainly relying on rhizobium, and when growing in competition, it was recruiting other taxa. So at this point, how to use this information for developing a new crop-wheat interaction research? With the Ecology and Evolution Group at the University of Copenhagen with Paul Neve, right now we are focusing on some experiment using winter wheat and blackgrass as model species. The first experiment already concluded. We are waiting for the NGS analysis of the rhizosphere microbial community. We wanted, first of all, to characterize the rhizosphere microbial community. So to see if we test different cultivars of winter wheat and different population of blackgrass. First of all, if winter wheat and blackgrass have a different microbial community specific for species when they're growing in monoculture, and what is happening when they're growing in mixture. We kept the soil from which we grow the plants, and the next year, October, September, we will repeat the experiment, also testing the soil legacy effect. So if the root exudate released in the soil can have some effect, also shifting the microbial community. And last but not least, we are also checking, there are many other things that we are investigating, but these are the main research that we are developing. The effect of biological on different plant species. And that's because biological are used mainly by the private industry and the farmer for increasing plant growth, health, and fitness. We want to know if these biological are one fits all, so they are the same for all the plant species, or if they could have a different effect on plant species, and maybe also shifting the opposite, having a better effect for weeds than for crop. And with that, I finish my presentation, so I will be...
Thank you very much. We have time for a couple of quick questions.
Do you know what herbicides are doing to the microbiome? For example, herbicides which affect microorganisms like, polyuria or...
Yes. Yes, but what's the question? Sorry.
How they affect the biome?
Well, I would like to argue that all the conventional systems have an effect on the rhizosphere microbiome. Already using the fertilizer, synthetic fertilizer, we are going to include some external compound. And so this research should be for sure developed, if possible, comparing a conventional and more sustainable system without the use of chemicals, synthetic chemicals.
No, but herbicides affect the weed, the competitor. They are not affecting only the microbiome. They affect also the species, the plant species.
Okay, then
Taking place in this interaction. How this comes up with the yield or with some effect, biological effect.
I don't think that I got the question.
But I think you didn't use any.
No,
You apply herbicide.
No, I did not.
No.
I did not.
You kill,
Oh, you.
and you kill Dalopicus or you inhibit Dalopicus. How this will affect the microbiome on wheat and
Yeah.
other species.
Yes, sure. Maybe I didn't explain clearly. Our aim is to investigate the effect of biologicals on plant competition, not using herbicides right now, not using any external input, just to check how the plants were competing. They can affect the rhizosphere microbial community.
Well, just one very quick question.
When you're looking for the effect of biologicals, you have to distinguish between the interactions, between plant-plant interactions and the following consequences for the microbiome, and the other way around. There might be microbiological interaction that changes the competition between plants. How to distinguish between the two? You get my question?
Yeah, well, that's why we're growing. Now, could you repeat, please? Could you?
So you have plant-plant interaction.
Sure.
For example, one plant is quicker take up nutrients, and that changes the microbiome as
Sure.
a consequence. But it's not a driving factor to change the microbiome. It's just a consequence.
Well, that's it.
Looking for biologicals, you want to have a driving factor that change the microbiome to
Okay.
improve
Well,
the crop.
I would like to say that in the first is the natural ability of the plants to use microorganisms in the soil, and that is well, but still here what we want to test it is more the fact of how the biologicals could go out of target. So instead, improving and helping only the crop can go affect other plants that are growing in the same environment. How to discern between the two? No, I did not think about it.
Okay, thank you very much. We're going to move to the next speaker.
Yeah, so let's welcome Serap Sute, who will talk about Amaranthus palmeri in corn and cotton, which is quite difficult to manage and also interact with this crop.
Okay. Slide, okay?
Next slide.
Yes.
Good afternoon, ladies and gentlemen, dear Chair. Today I want to talk about a new wheat species in Turkey, which is actually first noticed in 2014 and recorded to the Turkish flora in 2016. You can see here the distribution of Amaranthus palmeri in Turkey. That's just in our studies in Asian region, very close to here, and also in Mediterranean region. We have this wheat also actually in southeastern Anatolia region, but we haven't worked on the populations there. Because of that, I will just present these species. Yeah, we did some research in the past since we have noticed this wheat species, and we have tried first some dose response experiments in Mediterranean region, and we try to find out if there is resistance or not, because there were many farmer complaints about inefficiencies of glyphosate. As the result, we found actually no distinct resistance case. It was less sensitivity, but not resistance case. But Professor Mennan has worked also molecularly on it, and he has found something in among these populations, some differences. And we have also worked on management studies in citrus orchards with different herbicides and mechanical control and so. And after finishing this studies, we have started with a new project which is supported by Turkish Council for Scientific Research, aiming, and is still going on, and aiming mapping in Asian and Mediterranean region. I have shown already the map. And the competition with different crops in the region where it occurs So in this case, cotton, maize, tomato, and also sunflower. I will present here some results from cotton and maize experiments. Yeah, and we have also worked on allelopathic potential of this weed on these crop species as well as in other species to show the role of allelopathy in competition and to show the role of allelopathy in invasion. And we have also worked with chemical control, and as the result it yielded with 5 master theses and 2 PhD theses, which are still going on. Okay, I just want to mention some methods we have worked with. We have carried out pot experiment and also field experiments. I will present just the results of pot experiments here due to our limited timing. And the competition was investigated at early and late competition timing. And for early competition, we have seeded the seeds of wheat species together with crop species at the same time. And for the late competition, first crop seeds were sown on the pots, and when they reached up to 6 leaf stage, also Amaranthus palmeri was sown, and the competition, we had also different densities, 1, 2, and 4 plants per pot. We grown single Amaranthus palmeri plants as well as single crop plants for the comparison. You can see here early stages of experiments. You can see the cotton and the weeds. They were emerged nearly together here, and in the late competition, the plants were so. The cotton and maize plants were 4 6-leaved stages, and the Amaranthus palmeri emerged later. Okay. I just want to show here the dry weeds, and in the early competition, we can see that Amaranthus palmeri reduced the cotton growth by about 90%, regardless of the density. 1, 2, and 4 plants, there were no differences. All suppressed the growth of Amaranthus palmeri, so by 90% into 2020, and around 40% in late competition period. It means Amaranthus palmeri suppressed cotton growth at both period, but much higher at early growth stage than the late growth stage. Similar results were observed in last year. It was around 80% reduction, regardless of the density again. And in the case of early competition and in the case of late competition, that it was about 20–30% reduction. Cotton affected the growth of Amaranthus palmeri also because it's a competition, and all plant has effect on each other. And we have tried to measure also this one. And in the case of early growth stage or early competition, we have just seen insignificant reductions. But together with the intraspecific competition, mean Amaranthus palmeri growth was reduced significantly in both years. But when we allow the crop, when we keep the crop free of weed until the 6-leaf stage, for 6-leaf stage, then after when Amaranthus palmeri emerged after that, we can see that Amaranthus palmeri has also, late competition. Amaranthus palmeri has also significantly suppressed, cotton has also significantly suppressed Amaranthus palmeri at late competition period. It was about 50–90%, depending on the year and density. Yes, in the case of corn, we have also similar results. Amaranthus palmeri suppressed the growth of corn significantly, regardless of the density again. and it was about 50-80% reductions depending on the early competition, depending on the density. And at later competition period, no significant reduction could be observed. And last year, there was no significant reduction in the case of Amaranthus palmeri and maize competition. And corn suppressed Amaranthus palmeri also very effectively, so significantly, and we can see that there were around 30 to 60% in 2020 and 2021, respectively, the reductions, growth reductions. And mean dry weight of the wheat was significantly reduced by corn plus intraspecific competition. At the late competition, maize suppressed effect of corn on Amaranthus palmeri. Corn suppressed growth of Amaranthus palmeri significantly at the late competition because the corn is tall plants and they shaded the later emerged Amaranthus palmeris, and it's a normal result by 90 to 95%. The Amaranthus palmeri was suppressed by corn at the late period. You can see here the greenhouse in experiments. Okay, in conclusion, we can say that Amaranthus palmeri has been newly introduced to Turkey and spread rapidly among crop fields and regions. And our results here showed that the wheat limits both of the crop, growth of the crops significantly. Especially early competition caused considerable growth reductions in both crop species, so early control is quite important. Yeah, cotton seems to be more sensitive to competition than corn, so that even late competition can cause significant reductions in cotton, based on the conditions. Therefore, the management of this wheat is important to reduce yield losses as well as to prevent the spread. Yeah, I have mentioned that we have worked with also sunflower and tomato at the beginning, and we have similar results also with this crop species. Our previous studies showed that some of registered herbicides can control Amaranthus palmeri effectively in corn, but not in cotton. There are some herbicides which can be used to control Amaranthus palmeri effectively in corn, but not in cotton. Yeah, based on grower statements and our own observations, we can conclude that the mechanical control is not always effective or feasible due to new emergencies and also rapid growth rate of the crop, of the wheat species. So chemical options with reduced residual herbicides seems to be more effective to control of this wheat. Yeah, however, relying on just a strategy relying on herbicides can have some side effects or some disadvantages, and because of that we should work on the integrated management of Amaranthus palmeri to have efficient control. Okay, my last slide is that the first author of this presentation is Mrs. Serap Sutay. She has carried out all these experiments in 2020. Yeah, our applause for her. Yeah, we have lost her in 2020, not because of Corona, but from other reasons, so very suddenly, and we couldn't believe that. Okay.
Thank you so much.
Questions? I'm sorry.
No, no worries, no worries. We have time for a question if you want. Yeah.
Do Amaranthus tuberculatus and hybridis occur in Turkey too? First question. And second, can anybody explain why gene amplification plays a role in the other Amaranthus species but not in Retroflexus?
Yeah, the first one, I don't know. Yeah, we have many new Amaranthus species in Turkey which are not so clearly identified. I guess yes, but I don't know exactly. And your second question can maybe Hüsrev answer. You asked for the gene amplification.
Games has shown that gene amplification leads to resistance. So, but you don't find it in Retroflexus as far as I see it.
No, we found it in Amaranthus palmeri.
Yes.
And actually, tuberculosis was introduced 1 month ago in Turkey. And also Amaranthus palmeri came to Turkey from South America. And 5 years ago, Turkey started to import some real animals from those countries. And then suddenly the Amaranthus palmeri was introduced in Turkey. And we had some experiments and we found that the resistance index for the glyphosate is equal to 4. So it's almost resistance because we never used the glyphosate against Amaranthus palmeri in Turkey, but we found that the populations are resistant. And we found that the mechanism of the Amaranthus palmeri resistance to glyphosate is gene expression.
That was not the question.
But we didn't find it in Amaranthus retroflexus, and I don't know what's the reason, to be honest.
Was that the answer of your question?
Yes.
Okay, then.
It was no answer, but
Then I misunderstood your question, sorry.
So thank you, Mehmet, and give a round of applause to Mehmet and Sarah also.
So our next presenter would be Stéphane Cordeau from the INRA in France, because the first presenter couldn't make it.
Yeah, thank you so much, Barbara. So, as you can see, I'm not Ines May. She was not able to come, and so I'm presenting on behalf of the team. And so this talk is about allelopathy, and particularly, we published recently a review where we were looking at field-based evidences of allelopathy only coming from living crop, and also studying only studies where different variety were compared. I will explain after, but just to focus right now that this talk will not deal with allelopathy due to crop residues and only living crop. So yeah, in the context of reducing herbicide use, we are looking for weed suppressive crop or cover crop to suppress the weeds, and the weed suppressive mechanism mainly rely on competition, which is a widely studied mechanism, and also allelopathy. There is a lot of studies on allelopathy in the laboratory, but the field-based evidences are quite scarce, particularly when dealing with living crop and cover crop. So here the objective of this study was to look at field-based evidences of weed regulation by crop allelopathy, which are independent from competition, because as you can see when you look at the mechanisms, competition and allelopathy through living crop occurs when the donor plant and the receiver plant are closed in the environment, and so there is this compound that are in the soil, but also the nitrogen and the water, and so this proximity can, at some point, introduce some bias in the identification of allelopathy. So we run a systematic review of the literature by using different keywords, and we were able to produce an exhaustive, reproducible and quantitative analysis of the corpus. We found 523 articles talking about allelopathy, and we removed some of them because, as you can know, allelopathy is also a matter of bacteria, fungi, etc. So we only wanted to work on weeds. Then we remove all the paper looking at crop residue to focus only on living crop. And then we remove all the paper dealing without any field experiments, and you can see that we lost about 80% of the paper from there to there, meaning that most of the paper were based on field on lab experiment. We then look at all the remaining paper, look at the abstract, the material method to see which one could be really analyzed and dealing with competition. And so we only identified 25 paper dealing with allelopathy and competition. Some of them did not mention any data about the weed pressure, the weed biomass, the weed density. And some of them did not study the competition, and because the focus of this systematic review was to try to find some field-based evidences that try to disentangle the allelopathy from the competition, we only deeply analyzed these 25 papers there. So these 25 papers were published from 1994 to 2020, mainly on rice and wheat here. And as I said, we focused on paper comparing different variety of the same species, because we hypothesized that within a species you can have a gradient, obviously, within the different variety for competition traits, and we would like to see if there is differences in the expression of allelochemical within the species. And so there is an average of 8 variety compared per species. The other mainly measure above ground variable. You can understand why. It's more easier to measure than the root growth, for instance, but we can see here, even if this kind of variable are quite interesting when you look at competition, very few of them were measured, ground cover, leaf area index, etc. When the author measured some stuff on the wheat, there were mainly wheat biomass and height. And the thing that is quite interesting, and at some point I think scary a bit, is that the paper dealing on allelopathy, for at least half of them were no measurement at all of allelochemical or compound in the soil, either in laboratory, either in greenhouse experiment, either in the fields. But when some of them did some measurement of allelopathy, they did that with different kind of experiment in the lab or the greenhouse. So we classified the author conclusion based on what they said in the abstract or the discussion of their paper, and so we can see that 5 studies among the 25 mentioned that allelopathy alone is responsible for the differences in weed infestation between the crop varieties. 13 paper mentioned that at the same time allelopathy and competition are at work, explaining the weed infestation. Only 1 paper mentioned that there is no allelopathy and that competition alone was responsible for the differences. And 3 papers, 6 papers in total, mentioned that it was quite difficult to conclude that allelopathy was at work or allelopathy and competition. But when we analyzed their discussion and how they interpret their result and what were the measurements they were analyzed in the paper, we found that most of them didn't measure any soil coverage by crop variety or competitive traits at early stage, or some competitive traits were highly correlated to weed infestation, meaning that when we look at either the design or the results, many of them were not able to precisely mention that competition was not at work in their experiment, and particularly some of them precisely mentioned that in the discussion, otherwise they disregard this point in the abstract. So we conclude, based on the analysis of the design, the measurement, and the discussion of the paper, the author, that only 5 and 2 studies, so meaning 7 studies among the 25, provided evidences of the presence of allelopathy, and 1 paper provided evidence of the absence, and we were completely agree with the other one that had some difficulty to find some evidences. So this paper, again, only focusing on living crop and only focusing on studies comparing different variety among 1 species, did a systematic review, and we clearly found a misuse of the term allelopathy, particularly because some of the paper were not able to disentangle the effect of allelopathy and competition. So I think that we need to be cautious when we look at this paper on this topic, because we all know that it's quite difficult to observe in the fields, and all the field evidence could be very interesting, but obviously we need to show that there is no competition that works. There is very few articles showing convincing evidences that of the presence or the absence of allelopathy, even if many farmers are asking about how to use this lever in the field to improve weed suppression. And in most of the paper, and we provide also in the review paper some path to study the allelopathy. The most convincing evidence were provided when different technique, different method were used, and when the results were consistent. So I think that there is more room for study on allelopathy, particularly on crop residue, which is a well-known mechanism of allelopathy, and which was not included in this topic. And I would like to thank you for your attention.
Thank you very much. There's time for questions. Yes.
Stéphane, how worried were you about publication bias? That authors who don't find an effect are less likely to write a paper and submit it. Do you know if there's a way in systematic reviews to account for that?
Yeah, there are ways to account for that, particularly to analyze the bias of publication due to the journal, due to the region where the publication were made. Basically here we take all the paper we had to remove all the one which was not in the scope, and we ended with 25 paper, and we deeply analyzed this paper, and we think that there are at least the 7 paper we mentioned at the end, quite good paper providing evidence of field-based evidences, but of allelopathy. But obviously there is this big bias that you can see that there is only 1 paper publishing and saying that there is no allelopathy at works, and there is this bias that sometimes you do not publish your paper when you do not, when the results do not support your hypothesis. So obviously, we are missing some of this study, which are not published, I think.
Excuse me. I've just been told that there are some microphones, like on some of the seats near you, so use them. Yeah. Thank you.
I think. Did you find valid and solid methodology to evaluate the allelopathic effect on the field instead of in the lab experiment? Because I think that one of the most important lack is find a good methodology to evaluate the allelopathic effect on the field condition and not in laboratory condition.
The people from online say if you could repeat the questions because they cannot hear the people.
Yeah.
Like for all presenters.
Yeah, so the question was about if we found some methodology to assess and identify the allelopathy in the fields, because this is where we want the allelopathy to be at work. And yeah, so in the review paper we provide some methodological way to assess this in the fields. Some of the paper, of the 7 paper, have done that. Here on the scope of this review, the idea was to try to disentangle the effect of both mechanisms. So, meaning that when you're sure that there is no competition for light, nitrogen, water, and there is still a decrease of wheat biomass when specific traits regarding allelopathy, like the amount of allelochemical compound in the soil increase, something like that, you can say that there is allelopathy. But for that, look at the paper from Agroscope, from Judith Wurst and this group. It's quite difficult because you need to separate the plant and the above ground biomass. They need to remain on the same spot regarding their roots to change allelochemical. You need to put enough water and enough nitrogen to ensure that there is no stress. But because of that, there is a huge biomass that you need to separate. And so we can do in the fields, so at least using the soil, because here is the idea to identify the allelopathy. One of the convincing tools was to use the soil of the field and to put some water into one big pot and using the water coming out from the first one to go in a second pot where both species are separated but using the same chemical compound going from one soil to another. So it's going far from what we call a field, but at some point this can be outside, this can be using the soil, and at least to identify the allelopathy.
Thank you.
No other question? Okay, thank you.
So now we welcome Alessandra Virili, who will talk about wheat diversity, mitigating wheat competition in lettuce.
We don't have to repeat.
My name is Alessandra Virili, and I will be presenting today some results on a trial focusing on the effects of different weeds and wheat diversity on the competitive ability of romaine lettuce. This is part of my PhD thesis investigating the weed-mediated agroecosystem services in vegetable cropping systems and their competition with vegetable crops. So, to give you a bit of a background that has led to the development of this trial, some of the objectives of the Common Agricultural Policy for the next years are, for example, to take action against climate change, care for the environment, and preserve our biodiversity and agricultural landscapes and natural landscapes, of course. At the same time, there is increasing evidence supporting the benefits of non-crop biodiversity. For example, this review article, I apologize, this review article about the mitigating effect of wheat diversity on crop yield losses and supporting results on different weed-mediated agroecosystem services in arable cropping systems. Focusing on horticulture, though, there is not much evidence on this topic, and non-crop biodiversity is still negatively perceived because it is thought that weeds bring crop pests and decrease the market value of vegetable crops, despite the fact that in other arable cropping systems and in vegetable cropping systems, non-crop diversity has been shown to have many positive effects, especially when we think about biological pest control and support of pollinators and beneficial arthropods. So the knowledge gaps currently are that there is a scarce knowledge on the ecosystem services provided by weeds in vegetable cropping systems, and that there is also little information on the response of vegetable crops to weed presence in general, and more so to weed diversity, as opposed to the literature that can be found on cereal crops. I would also like to spend a few words on the concept of weed diversity versus weed identity. So it has been demonstrated that species-rich communities exert less pressure on the crop than species-poor ones. Despite this, it has also been found that species-rich communities dominated by competitive weeds can exert the same pressure on the crop than species-poor ones. And this all relates to the characteristics of both the crop and the weeds. And some traits that we can consider in this case are, for example, plant height, grime strategy, so is the species competitive, ruderal, stress tolerant, the affinity to soil nutrient conditions, and whether the plant is a perennial or an annual, for example. So based on the information that we have and the knowledge gap, our objective was to assess the competitive ability of lettuce when grown with 3 different common Mediterranean weeds in arable fields and at the different diversity levels of these weeds. We hypothesized that weed-weed interactions would be higher at increased diversity levels. Despite this, we expected, given the nature of the crop, which is lettuce, and the weed species, which I will introduce very shortly, that weed identity would outweigh weed diversity. So, moving on to the species, we have romaine lettuce, which is a very commonly grown vegetable crop in the Mediterranean, and it is grown all year round in open fields and in greenhouses, and it is managed quite intensively. And we have Italian ryegrass, which is obviously a grass. It's an annual species, and it is known to be very competitive. And it can grow up to half a meter on average of height. Then we have white mustard, which is from the Brassicaceae family. It is a broadleaf annual, and it has a competitive ruderal strategy, and it can grow to 1 meter of height. Finally, we have mallow, which is a perennial broadleaf. It is known to be a competitive species to grow up to 0.7 meters, but we need to consider that in its first year of growth, the traits related to competitiveness and height are not really manifested because its first year of growth is quite slow. The experimental layout is the following. So we had 1 lettuce plant per pot, grown with a total weed density of 10 plants. And the trial was replicated 3 times, 2 times in the spring and once in the fall. And it constituted of a randomized block design with 15 total mixture types, which, so these are the 8 mixture types with lettuce, and then just imagine the 6 mixture types without lettuce, you just take away the crop. So this is a little image of the experiment. Plants were grown in plastic pots in a greenhouse, and shoot and root biomass were collected at harvest of both weeds and the crop to calculate the competitive balance index of lettuce. Moving on to the main results, we can see that there was a significant interaction effect between mixture type and season on lettuce fresh shoot biomass, but this interaction was non-significant for lettuce competitive balance index, whereas only mixture type had a significant effect. Going more in detail, we can see the differences between the fall and the spring season. So in the fall, there are no significant differences between treatments, but in the spring, we can see that the differences are more exacerbated. Regarding the competitive balance index of lettuce, so when the CBI is negative, it means that the crop is less competitive than the weeds, whereas when the CBI is positive, it means that the crop was more competitive than the weeds. And we can see here that the CBI was negative only when lettuce was grown along with mustard, whereas it was positive in all of the other cases. Although we cannot see a clear trend of a clear effect of weed diversity, because if we look, for example, at these central mixture types, we can see that they have comparable means, but two have just one level of, are just with one weed species, whereas the other two have two weed species. Keep this in mind as we draw our conclusions. So, on one hand, it is positive that lettuce CBI was observed only when the crop was grown with mustard, although in all the other cases, a positive CBI is for sure a satisfactory outcome. It was apparent that mustard was our dominant and most competitive weed, and that the other two weed species could not completely outweigh the effect of this dominant weed. So maybe another similar trial with a different weed species, maybe more similar to the other two in height, for example, would be interesting to perform. Another positive result, though, that we found is that when lettuce was grown with the two most competitive species, so Italian ryegrass and mustard, its CBI was positive, confirming results found in zero crop, which have found that weed-weed interactions were higher than with crop interactions. Despite this, it seems clear that vegetable crops are less tolerant when they are grown with higher weeds, even in diverse weed communities, meaning that when we have these kinds of communities, maybe we should, if diversity conservation is a primary objective of the farm, maybe the local flora should be conserved and maintained at field margins instead of within the field, like we would do in zero crops or less intensively managed zero crops. This is all from me. If you have any further questions or curiosities, I will be here until Thursday. And if you want to follow the work of our agroecology group, you can find us on Instagram, Twitter, and Facebook at Go Agroecology.
Thank you, Alessandra. So we have time for questions if you want. Yeah. Can you use the microphone, because when you use it, this guy online can hear you.
Is it on? Okay. So, okay. A question regarding a subsequent crop. So, I was wondering, will you also consider that in your further works? Because you say now, for example, that Sinapis alba is not having a negative effect when competing with the Roman lettuce, but and that we should leave it at least on the field margins. But what about the subsequent crop? For example, if the subsequent crop will have a negative interaction with the Sinapis alba, and leaving the weed on the field margins, it is easily for it to enter the field again and to compete with the subsequent crop. So will you also consider that maybe in your further works, and then maybe be able to tell what crop should be subsequent to the lettuce when having some of the local wheat flora left there? Thank you.
An interesting consideration. So what I think is that these weeds have high seasonality. So for example, mustard, we started our spring growing season, went from March to May. And mustard, when lettuce was ready to harvest, basically, mustard was at its flowering stage. So I think there is also a good opportunity to manage the weeds in the field in a way that they do not then contribute to the seed bank, because it's all a matter of timing. And yeah, so I don't know if that, so I don't think in this case it would be an issue for a following crop.
Any other question? Yeah.
Yeah, this works. Could you maybe manage the competition of the weeds in a way that you find a competitor for, in your case, the mustard, that you could then grow on the field margins?
Okay, so I think when, in this case, mustard was the main issue, just because it is a very tall weed compared to lettuce, which is quite short. But mustard was chosen for this trial also because it is very common in wheat crop competition trials with cereals. So it was together with Italian ryegrass, so it was interesting just to see if we could transpose these weeds into vegetable crop systems as well. The way I would approach this trial in the future would be to not use mustard, but use weed species, if I grow lettuce again, a weed species that is maybe shorter, I don't know, always a brassicaceae, but maybe Capsella bursapastoris, something like that. Or use weed species that are comparable, that have one comparable trait. For example, mustard, amaranthus, and chenopodium. They are comparable in height with a crop that is also comparable in height. So I don't know, eggplant or maybe peas or beans, something like that. Yeah, hopefully I answered your question.
Yeah, thank you so much. So I think that there is a lot of questions on this topic because this is very interesting, but we will move to the next one. Okay. Sorry.
Okay, so our next speaker is Aude Ripoche. I hope I pronounced more or less correctly.
So good afternoon, everyone. I am talking about the effect of some crop practices like rotation and fertilization and their effect on the weeds. wheat community, wheat biomass, and also on the rice yield. And this was a work we done when I was in Madagascar with some colleagues from FOFIFA and also from CIRAD. Non, non, ça ne marche pas. Here? No.
This one.
No. Oops.
Okay.
Okay, I can.
Now we are at the second one.
So I can tell you some element of context during that. So we work in the Midwest of Madagascar, in the Vakinankarat region. It is just in the middle of the island. And we were at 1,600 meters of elevation. And we have, there are two main seasons, one dry and cold season from April to November, and one wet and and hot season from November to March. So the wet and hot season is the cropping season, the main cropping season. And during this season, we may observe until 1,200 millimeters of rainfall per year. So it's a relatively huge quantity. We are on fairly thick soils, which are really poor in terms of organic matter. And we work with really small farms, not bigger than 1 hectare, 0.5. In this region, there was a rapid extension and expansion of the surface of the area cultivated with rain-fed crops because the lowlands were saturated. And another big constraint for the rain-fed crops is that the farmers have no access to mechanization, neither inputs. So they are in the organic systems, but by default, because they cannot do in another way. And every practice is done by hand or with a spade. So we have we may observe a high pressure of pest, weed, and disease. So we have lots of things, white grubs, Striga, ME parasites with as Striga asiatica, blast disease, and various other insects. And recently, the fall armyworm arrived in the island. But weeding remained the most time-consuming activity on the farm because they don't use herbicides. So we want to know how crop diversity, even in a short rotation with one-fade rice, could affect weed infestation, and is it sufficient to reduce the weed impact on rice yield? So the main hypothesis was the fertilization is expected to increase wheat biomass and also rice yield, and so also with abundance. And rotation also could have positive impact on the wheat diversity, which could have no detrimental effect on rice yield and may support it. But rotation could and rotation could limit wheat biomass and wheat abundance, but wheat biomass and wheat abundance could have some negative impact too if wheat biomass was too big. So to study how crop rotation and fertilization may affect weed infestation, we use an experimental design with 4 randomized blocks, with four blocks for repetition, and we study rice monocrop versus and 2 years rotation. Rice followed by groundnut, rice followed by an association of sorghum and cowpea, and rice followed by an association of velvet bean and crotalaria. And we wanted to have some of gradient in the diversity, even if it's relatively small diversity. And in terms of biomass production, making the assumption that Velvelbin and Crotalaria was the most productive association to produce high biomass. And we combine this with two levels of fertilization, low one with organic manure, but it's a local organic manure of bad quality, almost 1% of azote in the manure. And a high level of fertilization with the same quantity of organic manure as F1 and mineral fertilizer to have around 1,070 units of nitrogen. And we make some measurement as floristic survey at the second weeding. It is just before rice flowering. With biomass measured at each weeding, there are two in each cropping season, and we measured rice yield. Every year, we have each element of the crop rotation grown. So you have the design just here. And then we also wanted to know how it was the effect on the harmfulness of the wheat community. So we use results from another work of a PhD student. He surveyed some farmers and asked them for their perception of harmfulness for 13 major wheat species. And we combined that with the weed abundance we observed, and we add the weed harmfulness at species level, but also at the community level during the sum. And after we made some classical statistical analysis. So in terms of wheat biomass, we observed some seasonal effect, and it was significant because some years affected biomass positively or negatively. And we also have an experimental design effect because each 2 years we had the same field for our observation. And then if we compare the level of fertilization, we also have a significant effect, not really huge, but significant, with higher biomass in the higher fertilization. And we also have a crop diversification effect, but only for, we have only a difference between the monocrop, rotation with groundnut and rotation with a sorghum copy had the similar effect and was different than the biomass observed in the rotation with the mixture of legumes, which were divided by 2 around. Then we want to know how the community structure could be modified. We observed more or less the same effect than on wheat biomass, that is to say fertilization increased wheat abundance, wheat species abundance, and the seasonal effect also were applied on community structure, and particularly in the third year, we had some species which disappear, maybe because we had a huge cyclone this year with lots of rain, and some weeds maybe did not support that. And then here we have the effect of the rotation on the community structure, and we can see that finally the species richness, was higher in the monocrop rotation, significantly higher in the monocrop rotation than in the mixture with legume association. So maybe it's because we are in the really short rotation, and it's not enough to impact wheat diversity, or maybe we have no other explanation for the moment. And we observe the same thing for botanical families. And after you also have the mean biomass for the record. So then we use this data on abundance to assess the harmfulness. So here at the left you have the harmfulness assessment by farmers for the different species. So 13 different species we had, and they cover almost 18% of the wheat community. Logically, as expected, Striga at the highest node because it's a really invasive and dangerous and harmful species for rice, for example. And then when we see the weed harmfulness at species level, we also observe an effect of the rotation because we had more competitive species under the monocrop than under the other rotation. For example, if you see Eleusine indica, the third one at your right, it was a little bit less important in some of the rotation. If we made the sum, we also see that the rotations tend to select some contrasting community level of harmfulness. And finally, we, as for weed biomass, we observed that the weed harmfulness seemed to be lower in the rotation with a mixture of legumes than in the monocrop. The rotation with groundnut and the mixture of cowpea and sorghum was intermediate. And then we want to know how it is related, how it was related to rice yield, and finally, considering harmfulness, it was not. It may have some explanation that the harmfulness was an observation, not on the field of our experimental field, but on based on the observation of the farmers. So maybe it may be it is a bias. And also, harmfulness is a combination between the ability of the species, but its functioning at field. So when we see with biomass, as Alicia this morning, we see a clear significant relationship between rice yield and wheat biomass. And for 1 ton of biomass observed at a field, we may lost around 2 tons of rice yield. So it has a huge impact. And finally, we also saw that under a low level of fertilization, that is the graph at the left side, so you have a significant impact of wheat biomass. But this impact may be alleviated according to the rotation. And we had less impact with the rotation with the association of the two legumes. Okay, at the right side, you can see that for the higher level of fertilization, we observe no impact on rice yield. You cannot draw the conclusion that weeds help rice to have an higher yield, but it was because everybody has enough to eat, sort of, because we have a huge impact of, we had a huge supply in nitrogen. But also you can see that the rotation affects the level of rice yield, and we also observed a significant effect of the rotation on rice yield in this level of high fertilization, and the rotation with the combination of legumes at the higher yields. Finally, in discussion, in a conclusion, so we see that as expected, fertilization increased with biomass, but it was not a huge problem for rice yield, because we also saw that the rice yield increased with high fertilization despite this higher wheat biomass. It was also because the rotation could impact this effect and the effect of wheat biomass. Finally, the more diversified rotation lead to a less harmful community, and when nutrients are limited in our F1 treatment, rotation play a key role because it allows to reduce wheat biomass and then to reduce yield loss. But we did not observe any increase in wheat diversity. So because maybe because we are on short rotation, but maybe because we have no herbicides and the seed bank is already huge. I don't know, or it's because competition between weeds. And finally, harmfulness remains hard to define. We had lots of work to do to better characterize this at field. Maybe use functional traits, and we had to decide which one, and characterize more tropical species, because there is a huge gap of knowledge considering tropical species, and we don't have lots of data on functional trait for this one. So it's a work in progress, and we do that in sugarcane currently. Thank you. Sorry for the time.
Any very quick question? Okay, thank you very much. So I'd like to thank all the speakers for adjusting to the time. We're sorry we're a little bit late because we started a little late. And just to tell you that posters 16 to 22 on the poster session are related to this session, so you can go and check them out also. So thank you very much.
Thank you.